I'd like to start by thanking you all for logging in today and thanking our guest speaker, Dr. Xiaoping Hu. Dr. Hu is currently a professor and chair of bioengineering at UC Riverside. He has served as faculty at the University of Minnesota, as well as within the, he was the professor in Georgia Research Alliance Eminent Scholar in Imaging for the Wallace H. Coulter Joint Department of Biomedical Engineering, that's at Georgia Tech and Emory. He has over 300 published peer-reviewed journal articles with over 27,000 citations. He is considered one of the early pioneers of functional mRNA and has been credited with many significant advancements to this specific type of imaging. He serves on the editorial board for Brain Connectivity and as an associate editor of Magnetic Resonance in Medicine. He is also a fellow of the International Society for Magnetic Resonance and a fellow of IEEE and of the American Institute of Medical and Biological Engineering. He is extraordinarily accomplished, and we are so very lucky to have him agree to come speak with us today. And my only sad thing is that it's a beautiful spring day in Alabama, and you aren't able to actually physically visit us. But today, Dr. Hu is going to share with us a talk entitled MR of Parkinson's Disease with Multimodal MRI. Thank you very much, and I'll turn the floor over to you now.
Thank you, MK, for the introduction, and I want to thank Jay and Carrie for the invitation. I know you guys have a very thriving department. When I visited last time, that must be like 2 years ago, right? Can't remember. So yeah, I want to talk about imaging of Parkinson's disease. I see I made a little typo. It should be imaging of Parkinson's disease with MRI, because this is one of our latest research efforts. It actually started out when I was at Emory, which is not too far from you guys. We still collaborate with the neurologist at Emory. So let me, let's see. Okay, here we go. All right, so just for engineers, I would like to say, for Parkinson's disease, there are two characteristics that we could try to sort of image, right? One is the degeneration of the catecholamine neurons. These neurons actually contain neuromelanin. Melanin is, sort of dark pigmentations that we can see on our skin, but you can also see these pigmentations in the neurons. These neurons are dopaminergic, and their degeneration, of course, leads to some of the symptoms in Parkinson's disease. So this degeneration is mostly seen in the locus coeruleus, which I will show you where that is, a small nuclei, and also substantia nigra, pars compacta. In addition to this loss of neuromelanin neurons, there's also an increase of ion deposition in some parts of the brain, including the substantia nigra and the other nuclei. So we can image these changes with MRI, and also we can use these areas that we identify with changes and look at some other measures of MRI. That's why I called this talk was the multimodal MRI imaging. So let's first look at neuromelanin. What is neuromelanin? Neuromelanin is basically a black pigment in these dopaminergic neurons. And if you look at the histology of a normal subject, and you actually see the dark spots in the regular histology slide showing you the pigmentation, okay? Now, if you look at Parkinson's disease patients, look at the substantia nigra, there is a loss of these pigmentation, which indicate the loss of these neuromelanin neurons. Okay? It has been shown that neuromelanin is a macromolecule that is paramagnetic, and also you can see this, as I showed you, you can see the depigmentation in the patients. And if we can detect this, of course, then we can detect Parkinson's disease. So before I delve into the how we can image neuromelanin with MRI, I just want to give you like 1 minute introduction to MRI for those of you who don't really, let's see if I have that slide. Those of you who don't really know MRI. So MRI contrast is based on strength of the NMR signal, and that is determined by how fast the NMR magnetization can go back to its equilibrium, so that we can sample the signal at different times. So this going back to equilibrium exponential growth, and it's characterized by a relaxation time known as T1 relaxation. So most of the images that we do use T1 is based on how fast the signal grows back, then we can look at it, we can probe it again. So for example, if you look at gray matter, it grows back slower than white matter. Then if you are imaging it, say, after 0.75 milliseconds, then you can see the gray matter is going to be darker than white matter, okay? Similarly, the signal also decays after we generate it. And that decay is again described by an exponential decay. Then how much signal we have is going to also depend on where we acquire the signal and how fast the signal decays. And this is known as T2 contrast. So MRI is basically a multimodal approach itself. And you can see that in clinical applications, we can see these here. So these are just standard T1 contrast image and T2 contrast images. And you can see, as what I showed you earlier, that the gray matter in the T1 contrasted image is darker than white matter. And also, interestingly, this tumor here shows up brighter because this region apparently has a shorter T1 because of pathophysiology here. In contrast, you can also use a T2 weighted imaging sequence that shows actually reverse contrast between gray matter and white matter because the gray matter has signal decaying faster, slower than the white matter. Also, in this part of the tumor, mostly likely, edema, it has a longer T2 and shows up bright. Of course, like most other imaging methods, you can also introduce additional contrast by exogenously administer contrast agent. For example, this is a contrast agent that produces a T1 enhancement. And you can see that the tumor now shows up also in the contrast, on contrast the injected image. And interestingly, the tumor shows a enhancement around the periphery of the tumor because that's where the mostly vascularized region of the tumor. So this is, routinely used in clinical imaging. I would not go much further into that here. What I want to tell you is that what we're using for imaging neuromelanin is a contrast similar, related to this T1 contrast, basically by reducing the amount of magnetization that we can probe into, okay? So this is initially used, proposed by this Japanese group led by Sasaki, who used a T1 weighted sequence to show that there's a faint, you can see there's a contrast for the substance nigra. It's a little bit brighter here. I hope you can see it on your computer in these images. Okay, you can also see the lucus coeruleus, which is very small dots, bright dots here near the ventricle here. So they used this sequence known as the turbo-spin echo sequence, which actually gives you a contrast that is known as incidental T1 magnetization transfer contrast. I will explain that in a minute. I know many of you are not MR expert, so I'll explain that in a minute. Okay, so later attempts to try to use a explicit magnetization transfer contrast, which I will also explain. And what's important is that it was found that this kind of contrast is localized to neuromelanin regions of the brain. So now I know there's a lot of students here. I want to talk a little bit about the principles behind this. Before I do that, I want to show you indeed it's not a simple T1 contrast. So if you look at a T1, a map of the T1 in the brain, you can see this is the substance nigra here, and there's not a lot of contrast that indicates the substance nigra pars compacta, which contains neuromelanin. Only with magnetization transfer, you can see this is a T1 weighted image. This is with magnetization transfer. Now you can see the substance nigra much more clearly. So this has been resolved, and our group has contributed to this resolution to make sure that we know that it's an MT contrast, magnetization transfer contrast, not T1 contrast. It is still unknown why this is a very bright contrast. We still need some more investigation there. But now I want to go back to talk a little bit about the principles MT. So if you look at the NMR spectrum, I think if you took organic chemistry, you would know your NMR spectrum. You will see that, in this, if we take a piece of tissue and look at NMR spectrum, there are really two major species. One is arising from the mobile liquid type of protons, mostly water. We know that body is 80% water. So this is mostly water, okay? So because the protons are mobile, then it gives a very sharp peak in the NMR spectrum. But also there's also macromolecules, including, I think, neuromelanin. Those would have a very broad spectrum because they are not so mobile. So then their NMR signal decays very quickly. Then when you perform a Fourier transform of something going down very quickly, you know that you're going to get a broad spectrum. So this is sort of fortunate in a way, is that we cannot detect this, this broad peak directly, but we can detect it indirectly by manipulating this broad peak. And through exchange, whatever manipulation you do here is going to be transferred to this observable signal here. So we can indirectly observe this signal. So this is known as magnetization transfer, basically by changing the magnetization in this broad peak here, and through exchange, this magnetization manipulation is going to be transferred to this signal here. So basically now reduce this peak. This would reduce the amount of signal that's available that's observable. So you can see this a little more clearly in this pictorial depiction here. So we are looking at the images, we're looking at the hydrogen nuclei in the water signals here, okay? These hydrogen nuclei would normally be lined up, then they would form a vector that we can perturb and observe, right? So these are the macromolecule. They also have these hydrogen nuclei, but they are not observable because the signal decay way very quickly. But we can still perturb these nuclei. So we perturb them. So now we reverse the direction. So they are, instead of pointing in the equilibrium direction, now they are pointing down. And then through exchange of the protons, some of these protons in the water is also going to be pointing down, like this one here and this one here. So because this is a partial sort of flipping of these protons, we're going to have reduction of the protons going up. This vector sum of everything going up is going to be reduced, okay? So our signal is going to be reduced. This allows us to get this, the effect of saturating this, and in the observed image, you can see the total signal is going to be reduced. So this is known as magnetization transfer. Now, how do we have this incidental magnetization transfer? In MRI imaging, we actually images multiple slices almost simultaneously. Okay, so when we image this slice in red here, we of course would apply the manipulation directly to this slice. Okay, so that's right on the resonance at the peak, at the center of that peak, right? But when we image other slices, we are going to be applying the manipulation off that peak. And if you remember this slide here, when we apply the resonance off that peak, then we're actually going to saturate the macromolecules. So basically, when we image these other slices in this bluish yellow, a greenish yellow, you can see that this is going to be saturating the macromolecules in this slice here. Okay, so this gives the incidental magnetization transfer effect. You can see that here. So if you only image one slice, there's no incidental magnetization transfer. Now, if you image more than one slice, you can see the signal slightly reduced, okay? And you can see there's also a contrast increase between gray-white matter because gray matter and white matter have different amounts of macromolecule content. And this effect also depends on the magnetic field strength, and most of the clinical scanners are either at 1.5 Tesla magnetic field or 3.5 Tesla. Okay. But this is beyond the scope of this talk. For the direct or explicit magnetization transfer is you actually saturate those macromolecules before you start imaging, directly or explicitly. Okay, so this of course has a better control. If you are doing this indirect saturation, then you're depending on the image of other slices, and you can see that the effect depends on how many other slices you're imaging. So it's not easily controllable, and it's not very quantitative. So we are arguing that what we really should do is to use this explicit magnetization transfer for neuromelanin imaging. Okay. So you can see that here, you can see the contrast with a multi-slice turbospin echo, which gives the incidental MT. You can see some effects, and you can see the neuromelanin here showing up as bright. But with the explicit magnetization transfer, then you can actually see it very clearly. Okay. So in addition to do this, we also introduced a few other measures or modification to the sequence. One is that, these pulses actually gives the patient a lot of radio frequency power or energy, just like almost like a microwave oven. Okay, so then we run into the danger of hitting the patients too much. So we actually have to reduce the amount of what is known as SAR, or specific absorption rate, the energy deposited into subject. Okay, we can see that with our approach we can reduce this. This is the turbospin echo sequence. This is the amount of power that's deposited to the subject, and this is our sequence without any special manipulation, just magnetization transfer. Then we can also reduce this further here. Okay. Let me change this so I can see the slide myself. Okay, so we can now, with this, you can actually then increase the number of slices you can cover in order to cover the whole brain, because the more slices you image, the more energy deposited into the patient. You may get into a limitation on the number of slices. So with this modification, we can now cover 16 slices instead of 5 slices. Sorry. One thing that also we introduced is to actually image, to specifically for imaging the leukocoelus. The reason that it is important to do that is that when we image this leukocoelus is very small. It's only 2 to 3 millimeters wide. I will show you in a minute what I mean. And if you have worked with MRI, you know that the MRI image resolution is not most of the image resolution order a millimeter. So if your structure is only 2 millimeters wide, it's kind of hard to see. So we actually have to go to sub-millimeter resolution. The problem with that is that when you go to high resolution, you spend more time to image, and then you also lose your sensitivity. Okay. So we have to acquire multiple images, the image multiple times, and average those images to gain enough signal to noise ratio. Okay. However, when you acquire multiple images of the leukocoelus is actually not still during the time you acquire these multiple images, because the leukocoelus is very next to the ventricle, to the fourth ventricle. The ventricle actually pulsates because it contains cerebral spinal fluid. It actually pulsates over the cardiac cycle. Okay, so when you try to get these images that of the leukocoelus, but at the quite different time, they're going to be slightly sort of moved from each other. So if you just put these images together, add them, then you're going to see a blurred version of the leukocoelus. So instead of just adding them simply, we actually try to align up these images up before we add the images together. So this is usually not done in MRI. So we actually propose to do this registration of the individual measurements before we add these measurements together. Okay, so this just shows you the contrast enhancement with an explicit magnesium transfer. A standard explicit magnesium transfer, you can see the contrast went up from 2.4 to about 3. So that's about a 20%, 30% increase. And we can reduce the amount of SAR or power deposition without sacrificing the contrast. So this is the protocol we're using. Okay. Oh, sorry. Let me see. Okay, that's better. Okay, so I can see the slides myself. Okay, so you can see now, this is a turbo spin echo sequence showing you the contrast of the local substantial Niagara pulse compactor. You can see compared with directed MT transfer, the contrast is lower. Similarly, you can also, now you can see how small the local solus, these are these little bright dots. And you can see that we are also getting, better contrast in our sequence. So that's good. Now let me just reorient you with the anatomy. So this is the substantial Niagara here. These are all around the brainstem, and this is the local solus. This needle-like structure. There are 2 of these, one on each side. They are about 2 to 3 meters in diameter and 10 to 15 millimeters in length. Okay, so we need to image this, and this you can see this is the histological slide showing the local solus, and it's right next to the ventricle. Right, so this fourth ventricle is pulsating, and you can push the local solus a little bit. Okay, so it tells you that there is a motion we have to worry about. To show you how effective this motion is, you can see this is averaging the images without registering. You can see this is a blurred version of the local solus, and we can sort of bring the local solus into focus by lining the individual measurements up and before adding them. Okay, you can also see that the volume of the local solus that is derived from these images is reduced with this alignment. Okay, because the movement that blurs the image and makes it appears to be much bigger. In order to also apply this clinical, we also have to see how reproducible these measurements are. And these are the correlations between 2 measurements. And we can see that there is fairly good consistent correlation between the 2 measurements in substantial Niagara measurements and also local solus measurements. This is looking at the contrast. So remember, this anti-contrast is sort of a semi-quantitative measure of the amount of neuromelanin in this. Okay, so we can see that the contrast measurements is also quantitative. Okay, so with that, now we can look at the pathology. We know that based on histological studies, in Parkinson's disease patients, up to 70% of the Niagara neurons are lost at the time they can be clinically diagnosed. And also the neuronal loss occurs more in the caudal section than moves up to the rostral section. Okay. So let's say we can see this loss. So here we're looking at a control subject, and this based on neuromelanin MRI, we can sort of segment out the region with the neuromelanin in the substantial Niagara. Then this is a Parkinson's disease patient. You can see the volume of containing the neuromelanin neurons is smaller. Okay. Similarly, we can see that in the local solus. So this is the local solus. These are local solus. We can see in the normal subject. You can see perhaps a hint of it in the PD patient. So there's definitely a reduction in that. So the statistics of this study in a number of patients, this is about on the order of 60 patients. This was done at Emory. You can see there's a volume reduction in the substantial Niagara. Okay. There's also a volume reduction in the local solus. This is quite statistically significant. The contrast change is not as dramatic, but there is some change. So we were very happy to get this result, but we want to be sort of more quantitative. So one thing to do is to now compare, to actually correlate it with clinical measures. So one of the clinical measures, I don't know if there's any neurologists in the audience, I'm sure you are fully aware of this UPDRS measure that measures a bunch of behavioral scores. So the higher the score, the more severe the disease. So we performed a correlation of our measures, the volumes of substantia nigra, positive compacta, and the volume LC. If there is a correlation of the measure with the disease severity, there should be a negative correlation. Okay. So we see that there may be a trend negative correlation in the SN volume, but not so much so in the LC. In fact, there may be a slight positive, but it's really not significant. So when my students first saw these results, they were, of course, worried. They were wondering what went wrong. They were nervous about showing this to me. I'm sure, some students in the audience would say, this result doesn't make sense. Should I show my advisor? Well, based on what I'm going to tell you, I would say, no matter what the result is, if you believe the result is true, you should show it to your advisor. So at first, when I saw these results, I was quite disappointed. I said, gee, it's good at discriminating between patients and normals, but it doesn't correlate with the clinical symptom. Are we really barking on the right tree? Well, it turns out it's not bad news. It's actually good news. Okay, here we go. Okay. It turns out, according to Brack, who published this paper in Neurobiology of Aging about the pathophysiology and also the staging of Parkinson's disease, he basically shows that, Parkinson's disease progression is going to be progressed from the lower part of the brainstem going all the way up. So you remember locus coeruleus here and the substantia nigra here. So locus coeruleus would actually start degenerating in early stages, then substantia nigra. So this is shown in this table here. CO is the locus coeruleus, and you can see locus coeruleus degenerating is already starting in stage 2 and 3 and 4, while substantia nigra would start in 3, right? So the patients that I showed you, that we studied in the data that I showed you, is mostly here because they have confirmed disease here. So there's actually may not be so much degeneration in these patient groups, okay? And at least less so in the locus coeruleus than the substantia nigra. So the lack of correlation in the locus coeruleus volume may suggest that they have already degenerated over here, but there's not so much further degeneration in this stage. So what does this mean? This means that locus coeruleus measure could potentially be an early biomarker. Of course, we don't have the data to show that yet because the patients that we started were mostly here, this stage here, not over here. Okay, so we are just starting a collaborative study with a consortium looking at people who have sleep disorders that may be early signs of Parkinson's disease, so that we can demonstrate this is really early biomarker. The importance of early biomarker is that, when drug companies develop a lot of these neuroprotective drugs, try to sort of stop or reverse Parkinson's disease, it is too late if they apply drugs to people in these stages here, because the degeneration has already taken place. There's not much you can do. It would be nice to be able to do to apply the neuroprotective drugs here to prevent further degeneration. Of course, they cannot do this test. They cannot even do a clinical trial in these people here because they do not know how to identify these people, right? So if we can identify these people, they can help them to do to test their drugs. Of course, if we can identify these people, they may provide a time point for these patients to be intervened so that the degeneration can be slowed down or stopped. So we're excited about this, but we still have to confirm our conjecture. Okay. So the next part I want to talk about now is the ion imaging. Remember, there's increased ion deposition in Parkinson's disease in certain regions of the brain. Fortunately, we can also image ion with MRI. Ion actually causes, ion is sort of paramagnetic, so it's actually causes a local variation of magnetic field, and then it elevates the signal decay. Remember the signal decay I showed you that, this exponential decay with ion, this exponential decay is going to be faster. We can measure that, and we could also measure the magnetic field changes caused by ion. Okay. So the way to measure the ion, this exponential decay is, of course, to get images at the multiple times along this decay curve. Then we can do exponential fitting to get the decay rates. The decay rate is known as the RT star, which is basically the inverse of the decay constant, time constant. Okay. So the presence of ion in the brain is very well known, even in normal subjects, right? So this is a histological slide showing that, you have ion in the substantia nigra pars reticulata. Okay, so remember our neuromelanin imaging was mostly looking at the pars compacta. So there's pars reticulata containing ion even in normal subjects. Okay, there's also ion in the red nucleus. So in fact, this property was utilized by people in the field to say we can use ion-sensitive imaging to identify the substantia nigra, although only pars reticulata has ion. So you can see that if we use T2 star-weighted imaging to identify, so this is a T2 star-weighted imaging that is sensitive ion, then we can see the substantia nigra pars compacta. But it's actually not very consistent across different subjects. So you can see maybe a little bit of that in the second subject here, but not much so in the third subject. So this was a practice maybe 15 years ago for looking at the substantia nigra with MRI. So because now we have the way to use neuromelanin, which sees the substantia nigra pars reticulata consistently in the MRI images. So the first thing we did is to see what happens if we now use both approaches to image the substantia nigra. Basically, we can do neuromelanin contrast shown here in blue and the ion contrast shown here in red. As I said, this is mostly predominantly in pars reticulata. This is predominantly in pars compacta. So there's actually not so much overlap. So the total overlap between these two is about 10%. This is in normal subjects. We want to first of all establish in the normal subjects, so we can derive an atlas representing both parts of the substantia nigra. So by using both contrasts, now we are having a more complete picture of the substantia nigra. So then questions, what can we use this for? Okay. We know actually most of the Parkinson's disease related degenerations happening in the pars compacta, shown in blue here. So we can now focus on this region when we use these measures of neuromelanin changes and also ion changes in patients. We can also use other measures. We can also now analyze other measures in this region. So the first example I'm going to show you Oh, this is just a 3D rendition of this to see that we can have a very nice picture of the subsystem Niagara. So the first thing that we did is actually to see if this region that we can identify now is both neuromelanin and also ion imaging. These regions can help us in analysis. So we first went back to the literature. There was a review article showing that even with diffusion tensor imaging, I don't know if many of you heard of diffusion tensor imaging, basically it's looking at how diffusion in tissue is anisotropic, particularly say in white matter. It's mostly going to be along white matter fibers. And that's a measure of white matter integrity. So people have applied that to subsystem Niagara and to see if there is a difference in that integrity in patients. So you can see that there is some difference. So this is the Z-score here. The Z-score is, average Z-score is about -1. So that's not very, statistically not very significant, as you know. Except for this one paper by David Valenko with a Z-score minus 0.5, 5.5 minus. The reason we figured is that most of these people are using a region of the subsystem Niagara defined by the ion, okay? So ion-sensitive MRI is used to define this region of interest, okay? And that's not a really good way of finding the diffusion tensor imaging differences because that's the region not so much affected by degeneration. So we analyzed, reanalyzed another group of subjects with diffusion tensor imaging. This is some data that we got from a German group. Then we see that, you know, if we use the neuromelanin defined region to look at the diffusion tensor imaging, and we can see a bigger difference. Okay, if you use just the ion defined region, then you don't really see much difference. So this shows you that by identifying the neuromelanin regions, we can zoom in and we can now see bigger differences in this diffusion tensor measure, right? Now we can also now look at the ion increase in these regions. Okay, so again, this is with a group in Hamburg. Okay, so they have a group of subjects with the, you know, this ion sensitive imaging data. We can analyze it. And the idea is now we have these masks, or they have these atlas, then we can put these atlas on the human, onto the patients, and we'll see. Also, we look at the ion depositions. The ion deposition showing up here in these dark regions here, and we can see how this ion deposition is overlapping with this blue region that is defined based on normal subjects. Okay, so this just shows you some patient data here. You can see in the control region, this is the amount of ion showing here in the control group. In the Parkinson's group, you can see an increased ion deposition. Then if you now overlap, if you now compare this with the neuromelanin, you can see that's this increase, which is below this region here, is mostly now in the neuromelanin area. Okay, so this can be seen also here. But the most interesting thing is that there's a huge difference. I wouldn't say huge, but the huge in the terms of the statistical significance. Okay, so if you only look at this overlap region, you can see the statistical difference is very high. The significance is high. In fact, the p-value is 10 to the minus 15, which is, you know, it's something that I don't actually believe, but at least it tells you the significance is very high. We also now did the correlation of this region's ion increase and with the UPDRS. Again, there's not so much correlation. Okay, there's not so much negative correlation. So this time, of course, we are not so disappointed because this may yet also indicate that this measure is another early biomarker. So this results is also now reproduced by other groups. Another group at Beijing Hospital in China who collaborated with us also show these differences in this in the ion deposition in the pars compacta. So that's very, very good, right? The final piece of data that I want to show you is to see how we can now map these changes in the neuromelanin change to some substructures of the substantia nigra. So I told you that substantia nigra contains pars reticulata and pars compacta, but there's also small structures known as nigrosomes. So here we have, let me see, we have nigrosome, this is nigrosome 2 here, and this is nigrosome, nigrosome 1 and nigrosome 2 here, okay? Nigrosome 1 is the largest of the nigrosome. And it turns out the degeneration happens early on in the nigrosome 1, okay? So up to 98% of neurons in nigrosome 1 are lost at the time of PD diagnosis, okay? Nigrosome 2 then has the second highest neuron loss. So this is nigrosome 2. Okay. So now we can use MRI images to identify these nigrosomes in the subjects, okay? So the nigrosome actually shows up as some bright regions in the T2 weighted sequence, okay? Then we can also look at the neuromelanin change in MRI. So here we have the nigrosomes in these patients. These nigrosomes are unfortunately not as visible in, this is in controls, and in patients it's not as visible because they have a lot of ion increase in there. But fortunately we can map this region from even the controls that we can now compare. Let me just move to the next slide. Okay, compare to see where these changes are. So this is the change. This is the reduction of neuromelanin. Okay, and then we can now compare these with the nigrosomes. These two clusters are shown to be one cluster corresponding to nigrosome 1 and another cluster corresponding to nigrosome 2, okay? So this is actually interesting to show that not only we see that there's changes in the substantia nigra pars compacta, but they are actually happening in these nigrosomes. I think that's, yeah, it's further shown here. So we can see this nigrosome 1 and nigrosome 2's losses, nigrosome 1 and nigrosome 2. So this was actually, recently published in Movement-Related Disorders, generated some interest in the field. So I think that's all the data I want to show you. Let's see, I've done about almost 40 minutes. That's great. So just to conclude, neuromelanin MRI can show differences in both LC and leukocoolus between PD and normal controls. And I also showed to you indirect evidence that the neuromelanin measured in LC could be possibly an early biomarker. We can also see the ion deposition mostly in the increase in ion deposition happens in this mostly in the neuromelanin area. If we just focus on that region, we can robustly distinguish between PD and the control groups, and the p-value is very high. And the correlation also shows that it's a possibly early biomarker, right? Further, reductions in neuromelanin sensitive contrast may correspond to a loss of melanized neurons in nigrosome 1, which also gives us a more specific pathophysiological change that we are imaging, right? However, currently we still have some unanswered questions. The first question is not known as to whether ion is causing the neuronal death or it is a byproduct. So we could use a longitudinal study using the approaches we developed here, perform a longitudinal study, then to see which happens first. It might be able to prove, to allow us to probe this causality question, right? The second question that I mentioned earlier is that our measure as possibly early biomarker still needs to be confirmed in patients, particularly improved normal patients, in a longitudinal fashion. Basically, we will study them before they are diagnosed with disease. Then if some of them turn into PD, then we can come back, look at these measures, see if they actually showed evidence of early onset of Parkinson's disease. So I would like to acknowledge my students and the collaborators, particularly Jason Lanley, who was a postdoc at the time. Now he's a staff person in my lab. Daniel Huddleston is a neurologist at Emory that initiated our collaboration. So we have been working together for more than 10 years now. These two are collaborators at Region Hospital in Shanghai, and this is another neurologist at Emory. There are other students which I didn't include here because there'll be too many of them here. So the other thing I want to acknowledge is, of course, the support for these studies were supported. These studies were actually supported partially by the Michael J. Fox Foundation. I don't know, some students may be too young to remember Michael J. Fox. He was the actor in Back to the Future. He actually was diagnosed with Parkinson's disease. Then he set up a foundation to support Parkinson's disease research. In addition, we also had the NIH support for these studies. I'll stop here. Thank you very much for your attention, and I would be happy to take some questions.
Thank you so much. That was just a wonderfully fascinating talk, and it had enough background on MRI that even I could follow, and I haven't done imaging in a very long time. I want to open the floor up for questions. I know we have several faculty members from both BME and other departments that are in, so if you would like to unmute yourself and ask Dr. Hu a question, please do so. Students, if you have a question, if you could pop it in the chat. I'm just trying to make sure we don't overload our speaker with too many people at once.
So I'll jump in. I'm Dave Standert. I'm one of the neurologists on the call. So there are a few of us out here.
Oh, great. I hope I didn't make a fool of myself in front of you guys. But.
Oh, no. It's a great topic. And, looking for Parkinson's biomarkers is really important. I mean, I'm struck by the locus coeruleus change. That's really dramatic loss of the signal there in these patients. And you make the point that, it's not sensitive to severity because it's probably completely lost at the early stage of Parkinson's disease. But, it's possible now to get cohorts of patients at risk for Parkinson's. We know how to look for these people with REM behavior disorder or genetic forms. It would be really interesting to look at locus coeruleus in an at-risk population.
Yes, that's a very good point. I mean, if you are interested in collaborating, we'll be happy to do that. We also have now a collaboration with the NAPS consortium. It's in North America, this REM sleep, basically REM sleep behavioral group, because these patients are rare, so we had to work with this group or this consortium to get enough patients. We just, the consortium just got a funded project. We have a piece of there to do the imaging. Certainly, I think we would welcome the opportunity to work with more.
Yeah, that would be really, the question I guess I have is, have you had a chance to look at change over time in any of these patients? All cross-sectional, but what is the rate of change?
Not in the patients I showed you. I did not include other data we have because that hasn't been published yet, but we actually, in a funded study by the Fox Foundation, they have a longitudinal study, and we actually have seen some changes in there. So it seems to be also have something to do with medication. There's a difference is also related to medication. I'll be happy to talk to you more about that in a separate setting. But,
no, that's exciting. Thanks.
Yeah, that's a very good point. Yeah.
So I had another question about the locus coeruleus, and these are beautiful, beautiful images. So it's historically also very difficult to get any kind of images of locus coeruleus because of the fourth ventricle. And what about functional images? Can you use some of the same kind of registration techniques for functional images for something like this?
That's a very, very good question. Yeah, we are actually working on that. So we have some success with functional images, not a great deal of success. And we also reviewed some, the limited number of papers they're claiming they have functional MRI, functional activation. And I think most of them are artifacts in our opinion. So that's why we haven't published our paper yet, but we certainly have used some of these techniques that I described. And particularly, we need to use a mask that's generated with the neuromelanin MRI to sort of zoom in on the activations. And the other thing is because of his movements, of course, there's some blurring, and also there's some susceptibility induced the shifts of the brainstem in the fMRI images that may be actually moving some of the signal people are seeing into the CSF region. So there are a lot of issues. I'll be happy to tell you more if you are interested in further.
Thank you. Yeah, that's good.
Yeah, I think we actually have.
We actually
have another study that's related to Alzheimer's, because locus coeruleus is also implicating Alzheimer's, and we are told that our grant for this project is going to be funded, then we can actually look at these issues. Particularly one part of that is looking at the functional activation. Sorry, I interrupted you.
No, sorry, I interrupted. I was just saying that we have a collaboration with your site that just got funded that we're going to be starting soon with Aaron Sites at UCR.
Oh, yes. Right, yeah. So Aaron and I are the PIs, are the co-PIs of this other grant on the aging. So yeah.
Yeah. So great. Anyway, if we can get locus coeruleus in those data, I don't know, that would be great.
Yeah. So Aaron is working closely with us on this. Yeah.
Xiaoping, thank you for the fantastic talk as always, and thank you for your our schedule on your busy schedule. You probably know this recent report from our Minnesotan group, CMRR, that look at the structure of the, compared to normal and schizo patient. The high resolution imaging show structurally the patient and volunteer structure are not significantly different. But the chemistry, the NAD plus ratio to NAD are significantly changed. And more severe, the schizophrenia patient, the more severe the change in NAD. Actually, they measure NAD pH. That's the phosphorus spectroscopy. So that's quite surprised. The structurally completely similar between the normal and schizo patient. And the chemistry is actually very dramatically different. That was quite a surprise. I wonder if you apply the same thought in your Parkinson patient, you may find some surprise finding also with the NAD, such as NAD plus NAD ratio or the other pH changes, for example.
That's a very good suggestion. Yeah, we are. Obviously, that's something we would consider doing. Right now, we're trying to get some low-hanging fruits. Hopefully, yeah. I would. I think especially in Minnesota, they have some high-field scanner that would make spectroscopy easier. Yeah. Thank you for the suggestion.
Wonderful.